so I've gotten more interested in the interaction of hemoglobinopathies or genetic variants of hemoglobin and Falciparum malaria. So I'll talk about that, talk about some of my own work, and like I said, mostly I'll talk about other people's work. It's hopefully not too general, but hopefully it is quite broad. One of the things that I think that is really interesting about this interaction between the host and the pathogen is that us being the host is that hopefully I'll try and tell a story which kind of integrates the epidemiological aspects, the clinical aspects, and some of the molecular aspects of that interaction, and talk about and discuss somewhat about how we can sort of leverage observations made at those different levels in order to assist malaria. I don't have any financial disclosures because I work in tropical diseases. My professional disclosures, I'm not a hematologist, but I'll talk about hemoglobin and red blood cells. And in general, first I'll touch on why I think it's important to think about and look into genetic susceptibility to malaria, not just as a sort of interesting intellectual exercise, and not because anyone's proposing to go and do bone marrow transplants on children in sub-Saharan Africa to protect them from malaria, but because it's useful as an almost natural experiment in order to better understand malaria's pathogenic mechanism. And then for those who don't think about malaria all the time, we'll talk a little bit about malaria, a little bit about hemoglobin as well, so you have some background information, historical aspects of thinking on the interaction between the two, and then some about its clinical impact. We'll touch on mechanisms, and then I'll probably run out of time long before that, but then touch on some unanswered questions, at least it seems. So beyond it being an interesting scientific and medical story, why is it interesting or useful or necessary to study genetic susceptibilities to malaria? Because this, I think, is the sort of, in a general sense, the chain of investigations which I think could actually help protect children either with preventive measures or therapeutic measures from Falciparum malaria. If in field studies and epidemiological studies, there's observations made there's a differential susceptibility to malaria in certain human populations, that can potentially illuminate mechanisms of pathogenesis that, for a variety of reasons, remain relatively obscure for Plasmodium falciparum. Once those pathogenic mechanisms are identified, one could hope to also use those to identify correlates of protection in patients with susceptibilities to malaria. And that, in an ideal world, could provide targets for vaccines and future drugs. And there actually are two case studies of this not being brought completely to fruition, but at least going down the pathway partially. As some of you may know, pregnant women in sub-Saharan Africa are uniquely susceptible to malaria, Falciparum malaria. Typically by childbearing age in sub-Saharan Africa, folks do not suffer nearly as much parasitemia with fever, so clinical malaria. But a unique subset of people who do is pregnant women. And that observation has been made, at least in the medical literature, dating to the early 20th century, and that observation is probably even earlier than that. But so there's, that is particularly true in primigravidate, women who are in their first-time pregnancy, and somewhat so in their second pregnancy. But women who have had multiple pregnancies are no longer susceptible to malaria as primigravidate. So this epidemiological field observation was made quite some time ago, back in the, I think it was the mid-90s. Pregnant women get placental malaria. Back in the mid-90s, it was determined that the red blood cells that are infected with malaria are adhering to chondroitin sulfate. The former Duke medical student who is now at NILD, not when he was a medical student, but discovered that interaction, which was a very important one for the field. And then later on in the beginning of the 21st century, I guess, discovered that finding of chondroitin sulfate A in the placenta was due to the elaboration of a parasite protein called R2-CSA on the surface of the infected erythrocytes. Subsequent to that, in the last 10 years, more data have emerged suggesting that antibodies directed against that particular antigen on the surface of infected erythrocytes protect women in their second and subsequent pregnancies from pregnancy-associated malaria. And so this is a correlate of protection from placental malaria in women who have had multiple pregnancies. And then there are ongoing efforts by a number of groups to design vaccines based against that or targeting that particular antigen on the surface of infected erythrocytes. Another case study for how this could work out in an ideal world is Plasmodium vivax malaria. This is probably the only time I'll talk about vivax, but although it is important. But for a long time, Plasmodium vivax is not, well, some would argue that it's not at all very much in sub-Saharan Africa outside of East Africa. But it's at least very, very rare in sub-Saharan Africa. And particularly in West Africans appear to have, for a long time, been known to be resistant to Plasmodium vivax malaria. In the I think it was the 1970s, in challenge experiments, it was evident that the West Africans lacked a particular antigen on the surface of their erythrocytes, which was essential for binding by the vivax parasites to the red cells. So the parasites could not invade the red cells, lacking this particular antigen that was absent in West Africans. More recent evidence has found that antibodies directed against that antigen on the surface of the parasite are associated with protection in children, and so there are efforts to develop vaccine against vivax malaria based upon this antigen that was uncovered based on the differential susceptibility to vivax malaria in West Africa. So in an ideal world, this can work out and provide promising targets for vaccines. So just very briefly about malaria, I won't beat you down with too many, but for those who either don't know or may have forgotten, it is a parasitic infection of the blood and affects the red blood cells. This is an actual parasite within a red blood cell. You can see the red blood cell looks pretty ugly and deformed. And here it is on a peripheral blood smear. Most commonly, you see little ring forms in Falciparum malaria in the erythrocytes. Of course, it's transmitted from human to human by Anopheline mosquitoes. Here is I believe that's a female Anopheline mosquito biting a human, and they like to live either in stagnant water or in pools of water and tires and things like that, depending upon the species. I will show you the life cycle later, because I think that's a surefire way to put people to sleep. But and as you know, it's an extremely large global health problem. This is one of a few maps that I'll show you. Malaria Atlas project in the last few years has put together actually quantitative maps based on mostly real data. This is not only a qualitative map showing you in all the shaded non-gray areas where Plasmodium falciparum occurs, but it's also a quantitative map showing how intense falciparum occurs. The colors denote the entomological inoculation rate, which is the number of infected bites individuals receive in a year. You can see that the darkest brown is over 100. So in areas, particularly in West Africa and northern Mozambique, individuals can receive over 100 infected bites a year. Understanding the intensity of transmission in those areas. So, like I put Gordian knot in the title for a few different reasons. It seems like a sort of simplistic view of things to say, well, geez, it seems obvious that if you get bitten by one of those mosquitoes bearing malaria and you're a human being, it seems like, well, then you're going to get sick, right? It seems simple. You just Plasmodium falciparum gets injected into you and you get sick. It's as simple as that. But there's quite a big difference between having a pathogen present in your blood versus actually getting sick. And the actual clinical manifestations of malaria, which are very protean, I would say, are dependent upon a wide variety of factors. Very elegant work pins it partially on transmission intensity, which alters the clinical epidemiology and clinical manifestations of malaria. Age, that seems like a pretty obvious one. Immunosuppression, nutritional deficiency or sufficiency, co-infected pathogens, parasite virulence, differences in strains, one's own innate susceptibility to the pathogen, as well as other infectious diseases. Adaptive immunity, which I'll talk about more in a few minutes as well, and malarial use. So it's not nearly so simple as just the pathogen and you get sick. And in reality, you can get sick in a lot of different ways. You can actually have asymptomatic parasitemia. We did a study in the Congo, in the Democratic Republic of the Congo, where fairly healthy adults answering, fairly healthy, I guess, that they had the ability to answer a 4-hour survey. So they were feeling pretty well, but greater than a third of them actually had PCR detectable parasitemia. So a large fraction of adults in high transmission areas will be walking around with asymptomatic parasitemia. Even children can remain asymptomatic. You can have mild or what's called uncomplicated malaria, which is a fever and you feel bad. and you have parasites in your blood. And then you can also have what, in this world at least, would put you in ICU severe malaria, which is most likely threatened for Falciparum malaria, and has a few different phenotypes as well. So it's clear that it's not just so simple as you get parasites injected into you and get sick. There are a lot of different contributing factors which result in many different clinical phenotypes. So why don't I mention the Gordian knot? It's a remarkable tangle of host factors and parasite factors on epidemiologic, clinical, and molecular levels. And so it's quite a tangle. So y'all have probably heard the legend, I guess, probably a legend of the Gordian knot. If you don't, I'm not a classical scholar too, I guess I should mention that. I know I was a biology major here too, so I don't think I took any classical history courses. But anyway, Alexander, supposedly one of the greatest military generals of all time, supposedly hunted Egypt in battle. He was, I don't know, I guess he wasn't technically Greek, he was Macedonian, eventually assembled an empire, especially from the Mediterranean all the way to India. This is in the 4th century BC. But anyway, he was when he was young, he assumed the throne at a fairly young age. I think he might have been a teenager, actually. And early in his military conquest in the city of Gordian, which sits in western Turkey, he came upon a mythic ox cart, which was affixed with the Gordian knot. It's a little unclear as to whether or not it was tied to a post by the Gordian knot, or if it was somehow disabled otherwise by the Gordian knot. But a prophecy had been made that whoever untangled the Gordian knot would go on to become king of Asia. That's obviously all apocryphal. But anyway, he was a student of Aristotle, or a pupil of Aristotle, in his earlier years, so I think he set about trying to untangle the knot, put his mind to work. Eventually became frustrated and took his knife or his sword and cut through the Gordian knot. And so it became a metaphor for a sort of an unexpected and bold solution to an intractable problem. So Alexander the Great, of course, that was in Gordian in 334. He went on to assemble his empire. Eventually died at the age of 32 here in Babylon after a sort of a subacute, about a 2-week illness marked by fever and decreasing consciousness, which, I think it makes an interesting story for people to say he was poisoned, but Babylon was also quite, or this area was also quite malarious at the time, and he had fever and decreasing consciousness, so there's obviously no definitive diagnosis for his death. Could have been malaria after all. There was another story. There was a New England Journal paper about 10 years ago which tried to pin it on typhoid fever based upon some reports of symptomatology. But anyway, so that was Alexander, and that was his slicing through the Gordian knot. And in a couple different ways, hemoglobinopathies, as we'll talk about, slice through this complex tangle of Falciparum malaria pathogenesis in order to protect severe malaria. Malaria, as Alexander, is hemoglobin. Very briefly on hemoglobin. Hemoglobin is the oxygen-carrying protein of our red blood cells. And in fact, the derived weight of the red blood cell, little did I know, it's 97% hemoglobin. So it's a remarkably prevalent protein. And a lot of actually, even for other reasons, a lot of early protein work and successes in protein biochemistry were accomplished on hemoglobin. I think it's because it's so easy to get a lot of it, because you need to get a lot of protein. I'll only tell you things that I think are germane about hemoglobin, partially because those are the main things that I know about hemoglobin. But you have two alpha chains and 2 beta chains. It's a tetramer of those 4, which combine to form normal adult hemoglobin, which is hemoglobin AA. And there are 2 main ways in which people can manifest hemoglobinopathies. You can either have a reduced quantity of one of these subunits, or you can have a reduced quality of one of these subunits. The reduced quality we call the Thalassemias. The reduced quality we call either beta-globin mutants, sickle cell trait hemoglobin C. So I'll tell you what the basics that you need to know in order to learn something about its interaction with malaria. Briefly, beta-globin mutations, and that includes, like I said, normal hemoglobin is hemoglobin A. That includes the other letters like hemoglobin C, hemoglobin E, and hemoglobin S, which is also called sickle trait. So you have this tetramer. If the 4 proteins come together, you form normal adult hemoglobin AA, and your red blood cells look normal. That's a normal blood smear. If you have a mutation at one single locus in one of the beta chains, you can end up with hemoglobin AS, so you have a normal A from one of your parents and an S from your other parent. And for the most part, you have a normal blood smear. You just have sickle cell trait that's fairly clinically silent. You have a second mutation at the same site in the other copy of beta globin that you received from your other parents, end up with hemoglobin SS or sickle cell disease, and you have obviously a morphologically very abnormal blood smear. And without sophisticated medical care, obviously substantially reduced lifespan. For the most part, I won't actually talk that much about hemoglobin SS. Hemoglobin SS is important for lots of different reasons, but when we're talking about malaria, I think that biologically the really important state is hemoglobin AS, which nature has promoted in order across sub-Saharan Africa in order to protect children. And children with hemoglobin SS, I think evolutionarily, medically they're important, but evolutionarily they weren't necessarily selected for because of its interaction with malaria. Hemoglobin C is a different polymorphism, same locus as the mutation that causes hemoglobin S, but a different substitution. You have a normal blood smear if you get one copy from one of your parents. If you get two copies from your parents, hemoglobin CC, again, you have an abnormal blood smear, but like the others, it's actually compatible with full life. It's fairly clinically silent. And I'll also, I may mention this again later on, but the remarkable thing, bear in mind that this is an extremely common protein. 97% of the red blood cells, of all of our red blood cells, are made up of this. But we're only talking about substitutions at one single locus in one single protein in our bodies. And so for all the effects upon malaria and upon other diseases and upon public health and medical care, it's only one single mutation, particularly for hemoglobin AS, which is a single mutation in a single combination. The Thalassemias, you can have reduced production of these two different mutations. You have two copies of beta globin, 4 copies of alpha. They normally assemble to make this tetramer. If you knock out one beta globin, you get beta thalamyr. Knock out both, not compatible with life, you get no beta globin mutations. You have substitution of other globin genes for a while, but it's not compatible with life. Alpha thalassemia, you knock out increasing numbers of copies of the alpha globin gene. These little alpha things denote your genotype, so a deletion of 1 here, deletion of 2, deletion of 3. If you delete all 4, that is also not compatible with life. And most of the clinical, or most of the pathophysiology is not necessarily because you're missing the ones that were deleted, because you have it imbalances the red blood cell. You have an overabundance of the unpaired ones, which you do possess. But anyway, knocking out, and these are, I should also say, these are, at least with beta thal, these are much more molecularly heterogeneous. So in different parts of the world, you have different mutations and different levels of alpha and beta globin produced. That's about all you need to know about the genes. So where do these things exist? You know, some of the earliest, we'll talk about Haldane in a minute, but you know, you can put maps of the distribution of the hemoglobin variants and malaria on top of each other, and it's pretty suggestive. This is a historical map of P. falciparum endemicity, I guess prior to any successful control measures, assembled from some data that these investigators at Oxford got from a Russian science project for the mid-20th century. But anyway, this was P. falciparum endemicity before anybody eliminated it. And then this is their data about the prevalence of the hemoglobin S allele. And so if you look at sub-Saharan Africa, you can see there's a pretty good overlap between the two, right? So that's suggestive. Hemoglobin C, same mutation, or different mutation, same gene. That's focused. The focus of hemoglobin C is in West Africa, some of those highly malarious areas of West Africa with the high EIRs that we saw in the previous map. And then alpha thalassemia. There's a few foci outside of sub-Saharan Africa, but at least the most common allele that's found in sub-Saharan Africa is also found in the malarious belt. And you can see it peters out down at the tip where of Africa where there's not so much P. falciparum. So the distributions are obviously very similar, suggesting that maybe the two of these things have something to do with each other. Obviously, one could argue that, oh gosh, it seems like malaria causes hemoglobin mutations. You could also turn this association on its head and say, well gosh, do hemoglobin mutations cause malaria? Which seems a little bit odder, but we'll talk about how that kind of might be there might be some truth to that possibility in a few minutes. So the Haldane hypothesis, a lot of you all might have heard the Haldane hypothesis. Haldane was one of the forerunners of modern genetics, I guess. And so the Haldane hypothesis has, at least in its general understanding, is that these hemoglobin mutations exist because they protect against malaria. And as near as I can tell, the Haldane hypothesis in print originated, as best as I can tell, from 1949. This was buried in a paper about the rate of mutation of human genes. He buried the lead here, although this is something that I think he's very well remembered for, among many other things. But he was talking about Cooley's anemia, which is beta thalassemia major in Greece. And speculating about some other things, he said, well, it's also conceivable that the heterozygotes are more resistant to attacks by the sporozoa which cause malaria, which was then prevalent in southern Mediterranean. He may have said this in other settings, he may have written it in other places, but at least in print, this is the first time that I can find him referencing this hypothesis that there's an interaction between the two. And yet it's kind of an aside in the paper that's something about a completely different topic. But anyway, that is the Haldane hypothesis. And this was in 1949. Other people may have been speculating on it at the time, and there were some initial reports in the 50s and 60s mainly from West Africa about the interaction between the two. But when I got interested in this about a year ago, I guess it was a couple of years ago now, I realized that a lot of the studies that were cited to support protection from malaria in hemoglobinopathies relied upon, I guess maybe, one would not consider modern epidemiologic methods in their clinical studies. Understandably, this was from an earlier time of medical research, but we decided to go ahead and take a look at and do a systematic review of exactly what evidence is there that the various polymorphisms protect from various clinical syndromes in malaria, and how quantitatively can we estimate the degree of protection between those studies. And so we published this, I guess, around this time last year, a systematic review of the clinical evidence. And for hemoglobin AS, you can see that the first study that met our inclusion criteria was 1967, but there was a long gap between then and 2001 before the next study that met our criteria was published. Once we meta-analyzed, these are all case control studies of severe malaria and hemoglobin AS, we generated an odds ratio of 0.09, suggesting 91% protection, which, because severe malaria is a pretty rare event, I think is a pretty good estimate of protection from these clinical studies. Uncomplicated malaria, we couldn't find much, at least from case control studies. We got an odds ratio of 0.30. I think that's probably an overestimate of protection, because uncomplicated malaria is so common in high transmission areas. And I'll talk about that more in a second. But 91% protection from severe life-threatening Falciparum Malaria hemoglobin AS was quite remarkable. And this is on a log scale, so these estimates look a little bit different. But if I remember correctly, there was no heterogeneity in the meta-analysis of these studies from various sites in East and Central and Western Africa, underscoring the fact that even in very different populations that possess the hemoglobin S allele, was still associated with this remarkable protection. Hemoglobin C has been slightly less studied, I think because its focus is only in one spot in West Africa, and it doesn't reach as high allele prevalence, I think for a variety of reasons. But there were several good studies just from the last decade or so with a protection from severe malaria of over 70%. AAC, so if you have one copy of hemoglobin C allele, only about 20% protection. There weren't that many data for that, but it seemed that as a pattern that we sort of expected. It's in proportion to the number of alleles that you have. And alpha thalassemia, either 2 deletions of your alpha globin genes or 1 deletion, was associated with mild protection, just over 30%, just about 20%. Again, proportional to the number of deletions that were present. Those were all case control studies. There are also quite a few nice prospective longitudinal studies following kids from baseline and then developing calculating incidence rate ratios, which I think is a lot more useful for trying to gauge the degree of protection from uncomplicated malaria, in my epidemiological understanding. But we came up with a 31% reduced risk of uncomplicated malaria with hemoglobin AS. Contrast that 31% with 91% from severe malaria, and you can see which sort of protected basket hemoglobin S puts its eggs in. Hemoglobin AC, not much protection in these studies, and nor was there protection in prospective studies for uncomplicated malaria for the Thalassemias. One thing that I don't have a figure to demonstrate is there's also a lot of studies that cross-sectional studies where they measure the prevalence of parasitemia in otherwise healthy, usually children and sometimes adults, and look at the proportion of people with hemoglobin mutations who had parasitemia versus those with normal hemoglobin parasitemia. And in those, roughly the same proportion of folks with or without hemoglobin mutations are parasitemic at any given time. And so if you put together the protection from severe malaria, the protection from uncomplicated malaria, and the lack of protection from what really is asymptomatic parasitemia, it really suggests that hemoglobin AS, and also to a certain extent CC, are really just protecting kids from from severe malaria. There's mild, if any, protection from uncomplicated malaria, and there's almost no protection from asymptomatic parasitization, suggesting that somewhere along the way it allows children to get bit, it allows the parasite to go through their liver and incubate there, be released into the bloodstream, and for parasites to be present, but yet it just prevents them from getting sick. So somehow it dissociates parasitization from getting sick, which I think is really interesting and important for reasons I'll try and touch on later. So to contrast these degrees of protection with other things that we do to protect kids from severe malaria, put it in some sort of context, the greatest malaria minds of the day have come up with these various interventions to protect kids from malaria. ITNs and insecticide treated bed nets. Meta-analysis suggested 45% reduction. Again, in clinical studies, optimally designed clinical studies with probably nicely working bed nets that have just recently been treated with insecticides. 45% reduction in severe malaria. Giving all infants intermittent preventive therapy with a drug that was active at the time in West Africa, 38% protection. Giving children intermittent preventive therapy with Banzidar. This was not meta-analyzed. These were just two studies that came out in 2011, but between 69 and 87% reduced risk of severe malaria. But obviously, all these things have downsides. Bed nets lose their insecticide effectiveness. The mosquitoes become resistant to them. They get cold. People don't use them properly. And these studies in which children were given infants and children were given preventive therapy, were given with a drug that remains active in West Africa, but for the most part is not, Banzidar, for the most part is not used in the rest of sub-Saharan Africa. And so animal drug resistance threatens the durability of these programs. And so to contrast those things that we do with nature's own protective mechanisms, homozygous alpha thalassemia, 37% reduction, hemoglobin CC, 73% reduction in children in West Africa, and then hemoglobin AS children all across sub-Saharan Africa, 91% protection from severe malaria. So it's almost like nature's public health intervention to protect children from severe life-threatening malaria, and better so far than anything we've dreamed of so far. I think I alluded to this a minute ago. Hemoglobinopathies impact the clinical epidemiology of malaria. There are a number of different manifestations, but broadly they're severe, mild, and asymptomatic. In normal situations, this is just a cartoon meant to be illustrative of the relative age distribution of these different syndromes. But severe malaria is mostly borne by very young children who lack, well, there's a lot of different reasons, but it's not because they lack adaptive immunity. Mild malaria is borne more by older children and younger adults. And then people of all ages can manifest asymptomatic infections, and we don't fully understand why people fall, other than age, why people fall into one category versus another. So if you were to represent this differently, if you took a population with normal hemoglobin, if this, again, this is just cartoon, if this represents all the children with asymptomatic infections, a fairly small proportion of those will progress to mild or uncomplicated malaria. And even smaller, severe malaria is obviously severe, but still quite a rare event in the grand scheme of things. This dot actually might even be smaller than that. But we really don't understand how and whether people make the transition from asymptomatic to mild disease to severe. There's speculation about it, but it's difficult to understand which host factors, which environmental factors, which parasite factors lead to going from this category to this category to this category. But what hemoglobinopathies seem to do, specifically hemoglobin A, S, and CC, is if you have the same pool of people with asymptomatic parasitemia, a much smaller fraction get mild malaria, but it's still substantial. There's only 30%, at least with AS, hemoglobin AS reduction in mild or uncomplicated malaria. And then a very, very small fraction of those go on to develop severe malaria. So it allows all these, it allows people with hemoglobin variants to be parasitized, but it does not allow them to get sick. And I think that those are, it seems to neutralize these critical steps to progressing from asymptomatic infection to mild or uncomplicated disease to severe malaria. Which I think is really important, and this is some of the vaccine makers, this is kind of what they're focusing on. It's known that in malaria endemic areas, you don't necessarily develop immunity to parasitization, so adults in endemic areas will continue to get parasitized. Like I said, in the DRC, it was greater than 30%. But for whatever reason, they just don't get sick very much. So this pattern sort of mimics how adults end up in endemic areas. And I think a lot of the vaccine design is really meant to be attenuating and disease controlling rather than sterilizing, promoting sterilizing. There's a couple of other interesting aspects. Well, I think they're interesting. Y'all might see if y'all think they're other interesting aspects of the protection afforded by hemoglobin S. So this is hemoglobin S exhibits hemoglobin S and alpha thalassemia manifest epistatic interactions with regards to protection from malaria. And essentially, alpha thalassemia modifies the effect of hemoglobin S on protection from malaria. What I mean by that is this. This is a very elegant paper published by Tom Williams and Company in Nature Genetics in 2005. They work in Kenya. They seem to have more patients and more data, at least in the malaria world. So probably only them could do this study. But if you look on the x-axis, see this is all patients with normal adult hemoglobin. There's a bunch of doubts, but if you just look at the triangles, that's severe malaria. This is risk ratio, incidence rate ratio of 1, right? These are children non-thalassemics, alpha, 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 and hemoglobin AA. With heterozygous alpha thalassemia, they have a reduced risk of severe malaria. Delete 2 copies, and then they have even more reduced risk of severe malaria. Then if you look at kids with hemoglobin S, kids with hemoglobin S with all 4 copies of their alpha globin genes present, I guess it's supposed to be 0, but they have some risk, but almost complete protection from severe malaria. That's kids with hemoglobin S and all 4 copies of their alpha globin gene. And when you delete one of those copies of alpha globin, they do have some risk. Some of them do get severe malaria. And then if you delete 2 copies, obviously these confidence intervals are gigantic because there just aren't that many kids with this genotype. But as you successively delete more copies of their alpha globin genes, in other words, give them alpha thalassemia, their risk goes back towards 1. So they lose the protection afforded by hemoglobin S in the presence of 2 deletions of their alpha globin genes. And I think that this is really, this is fascinating that the 2 traits seem to be in conflict. But I think it's also useful for lab investigations as well, because this is a very clear. This is in 05 that they published this, but this has been replicated on slightly smaller scales in other studies, and it seems to be a fairly consistent finding that alpha thalassemia antagonizes the protective effect of hemoglobin S. And so I think it's useful for laboratory studies as well, because it's a consistent pattern of protection that one can look for correlates of in vitro in laboratory studies. And this also I find really interesting. I mentioned before that you could turn the association on its head and say, well, do hemoglobinopathies cause malaria? Which sounds kind of ridiculous to begin with, but these folks, I guess, thought differently. David Modiano and Company in Nature Genetics just a couple of years ago published a paper looking at gametocytemia and the infectivity of children with hemoglobinopathies compared with the infectivity of normal children. So I avoided showing you a life cycle, but one thing that is important to appreciate, I think, is that for the most part, we've just been talking about asexual parasites In other words, dead-end parasites infecting red blood cells inside of us. A very small fraction of those will differentiate into sexual forms of the parasite. And it's those, these gametocytes, the banana-shaped things that you may have seen in pictures on blood smears, it's these gametocytes that are picked up by the mosquitoes, undergo sexual recombination, and they become diploid in the mosquito midgut, and then develop again into haploid sporozoites, and are then injected into the next person who's going to get malaria. So it's these things which drive transmission, and they're important for lots of different reasons. But anyway, there's some old literature suggesting that children with hemoglobinopathies have a greater prevalence and a greater density of these gametocytes than do children with normal hemoglobin. And we tried to look at this in some systematic fashion, but the data were just all over the place in a lot of the older studies. And because these things constitute such a low fraction of the total parasite burden, when you look under a blood stream, they're just hard to quantify. But more recently in this Nature Genetics paper, they did surveys, and they were working in Burkina, very, very high transmission area, and also a lot of kids with hemoglobin C. This is the gametocyte rate. Technically, it's the prevalence, I guess, of gametocytes, the sexual transmitting forms of the parasite. In the gray bars, kids with normal hemoglobin, and the red bars, kids with hemoglobin C, the one that 73% protection from severe malaria, only present for the most part in West Africa. See, all the red bars are much, much higher than the gray bars. So the kids with hemoglobin CC had far higher numbers, far greater proportion of them were carrying gametocytes at the time of the survey. And in addition, there's another, I think they also found that the densities were much greater in those children with gametocytes than the kids with normal hemoglobin. So it seemed like the kids with hemoglobin CC were carrying a much greater quantity of gametocytes. And they didn't have a nice picture to show you of the follow-up, which was also in the study, where they did a fairly elegant clinical design, one that I still don't quite understand. But they had children with different hemoglobin genotypes sleeping in huts overnight, and then they would collect the mosquitoes that fed on those children the next day. And they would keep those mosquitoes. They would dissect them a few days later and look for see which mosquitoes were infected, and they could also tell which patient the mosquito had bitten. And as it turned out, children with hemoglobin CC contribute a far higher fraction of the infective forms of the mosquitoes than did the children with hemoglobin AA, suggesting that these hemoglobin variants in the children were actually deriving malaria transmission. They not only had more gametocytes, more transmitting forms, but those were also being transmitted more efficiently to the mosquitoes, and leading to greater burden of mosquito infections, which then could be passed on to the next children. So it's actually, you know, we're talking about a parasite here, not a virus or bacteria. It's actually exactly what the parasite wants. If you kind of think about it, it doesn't kill its host very often. It's transmitted more efficiently by its vector to its next host. You know, the parasites don't want to kill us. We're not the definitive hosts of Falciparum. Like I mentioned, it undergoes sexual recombination of the mosquito. It really needs the mosquito, and we're just sort of it's sort of using us to get to the next mosquito. And so it's kind of exactly what the parasite wants.
You think the CC children are sort of using the mosquito as a stealth bomber for the other kids to eliminate themselves? Yes. They can have more parasitemia and survive, and therefore they can get more food if these others that can't eliminate them?
I guess that tends to be a byproduct of that dynamic. The kids with CC, I mean, and the kids with AS, they're at least in West Africa, they're almost like, with regards to malaria, they're like super kids. You know, they can almost completely neutralize malaria, or Falciparum malaria. Anyway, onward. So I think I've tried to convince that it's important clinically, it's important epidemiologically, and it's important for transmission. I want to touch some on mechanisms. I spent a long time making that picture in Adobe Illustrator, so I think I'll leave it up there for a second. This is the only, this is a diabolical illustrator. This is the only time I'll show you this is a very simplified version of the life cycle. It gets into red cells, right, and then it makes spherozoites, which then go on to infect the next blood cell. So anyway, I think that there are four broad hypotheses broad hypotheses for how these hemoglobinopathies attenuate malaria, which I'll talk about more in turn. But in general, they attenuate the parasite growth. There's a lot of literature on that, which I'll talk about in a minute. The growth in the erythrocyte stage. They can attenuate the specific pathogenic mechanisms of the parasite when it's inside the red blood cell. And there's a few of those. Infected red blood cells like to bind as endothelium, or the cells that line our arterioles and venules and capillary beds, and sequester there and cause pathogenic states like pregnancy malaria or cerebral malaria. They can also bind to uninfected erythrocytes and grow sets and clump up and block the capillaries. Like I mentioned before, the infected erythrocytes can bind to certain ligands present in the placenta. So there's it could be that hemoglobinopathies neutralize these specific pathogenic mechanisms. They can also potentially modulate the innate immune response. By innate immune response, I'm kind of I'm defining that very broadly to not only talk about effector cells, but also things like nitric oxide and hemoxygenase, angiopoietin, things, cytokines that I have very poor understanding of, that are obviously sort of an emerging recognition of how they're upsetting the balance of them can lead to disease states in things in malaria and otherwise. Or they could enhance the acquisition of adaptive immunity, like I think I mentioned before. Eventually, adults with normal hemoglobin will acquire at least disease-controlling adaptive immunity. So it could be that somehow hemoglobinopathies promote the acquisition of this. But this identification of this mechanism is really hampered by the fact that there's no known correlate of immunity, even disease-controlling immunity to malaria. There's no single, in other words, no single test that we can order to say, okay, you're immune, this person is not. So I'll talk about them, all these four broad hypotheses very quickly. There's a large body of evidence on restricting the growth within the red cell of the parasites in red cells with hemoglobinopathies. I think in vitro cultivation was first developed in the 1970s, and subsequent to that there were about 10 high-profile papers about how hemoglobin S, hemoglobin C, hemoglobin F, or fetal hemoglobin, alpha thalassemia, all in one way or another retarded the growth or the maturation or the invasion or the egress of the parasite in culture. This is just one representative graph from a publication back about 10 years ago. Hemoglobin CC cells. Those hemoglobin CC cells are the open dots, the closed dots are AA. I'll say that there are a lot of data on how these various hemoglobinopathies restrict growth. For most hemoglobinopathies, there's a corresponding paper that suggests that they grow normally. And having at least observed other members of the lab cultivating parasites in vitro, I know that they're very susceptible to, you can tune, the cultivation systems can be tuned either inadvertently or purposefully to produce a lot of different phenotypes very easily. Not purposefully producing bad data, but there's an extreme amount of variability from one place to the next. But I'll say that these data about growth being restricted in hemoglobinopathies, I think, were bolstered by a very important paper. I think it was published last year by two people in the room in Cell Host Microbe. That the Dursing reference is for his editorial from which this cartoon comes from, but not the actual paper, which was in the same journal. But at the risk of misspeaking about the actually empirics findings, they found that microRNAs, or short non-coding RNAs, that are present in the human red cell, their profile differs in red cells with hemoglobin S and red cells with hemoglobin. And so what they found was that these microRNAs that, like I said, differ between red cells are translocated or moved from the human red cell across the parasitopterous vacuole and the plasma membrane of the parasite into the parasite, and that these human microRNAs then bind to mRNAs, or transcripts of parasite proteins that are intended to be made into protein, and that alter the expression levels of those parasite proteins. And this, like I said, these growth studies go back more than 20 or 25 years saying the growth is different with this type of red cell or that type of red cell, but this was at least one of the first times that I'm aware of that an actual mechanism for the differences in those, like a molecular mechanism, not a cellular mechanism, but a molecular mechanism, was put forth to try and explain these differences in growth. And I'm sure if you have questions about that, Ashley, Greg would be more than happy to talk about it some more. So the second broad hypothesis is that hemoglobinopathies interfere with the pathogenic mechanisms of a parasite when it is inside the red blood cell. Down at the bottom here, this is a normal red blood cell. This is a red blood cell that is with normal hemoglobin that is infected with Falciparum, and this is a red blood cell that is with hemoglobin CC that is infected with a parasite. You can see it's quite different, and there are some other pictures that were not included to show that these all these little dots on the surface are sort of knobs composed of parasite proteins that are thought to allow the parasite inside the infected erythrocyte to stick to endothelial cells in the vasculature. Those knobs appear to be aberrant on the surface of infected erythrocytes with hemoglobin CC. And indeed, when in vitro, when you take cells with normal adult hemoglobin that are parasitized, try and bind them to endothelial cells, if they have one copy of hemoglobin C, it's reduced. If they have two copies of hemoglobin C, binding to microvascular endothelial cells is almost completely abolished. And similarly, rosetting, or the binding of infected red cells to uninfected red cells to form clumps of red cells, is also almost completely abolished in red cells containing two copies of hemoglobin C. And this same group has gone on to show that this is a phenomenon which is present in various degrees with hemoglobin AS, one copy of sickle trait, and hemoglobin F, fetal hemoglobin, and thalassemia as well. So it seems like it might be a sort of consistent cellular mechanism whereby hemoglobinopathies interfere with the ability to put these sticky knobs on the surface of the red cell and then adhere to the vasculature and cause disease. And in fact, this mechanism is also supported within, I guess it's slightly over a year now, by a publication by a group in Germany who produced beautiful pictures of infected erythrocytes. But essentially what it came down to was their I guess it wasn't their hypothesis, but their thesis ended up being that the parasite within the red blood cell uses the actin cytoskeleton, or the scaffolding for the red cell that is ours in our red cell. The parasite uses that and remodels it. Essentially, it's like a bird gathering twigs to make a nest. It uses our actin to make its own cytoskeleton in order to send proteins to the surface of the red blood cell. What they found when they used, that's over here on the left, and the orange lines are the scaffolding, the actin scaffolding. On the right-hand side is, I think this was a cell with sickle hemoglobin and hemoglobin C, so hemoglobin SC. You can see that there's a disruption of the orange lines of the actin cytoskeleton, and all these green blobs are parasite proteins that are meant to be exported to the surface of the infected erythrocyte. So I think it's possible that the hemoglobinopathies, for several different reasons, interfere with the ability of the parasite to remodel the actin cytoskeleton of the red cell, and thereby prevent the trafficking of parasite proteins to the surface of the red cell. This is innate immunity is another broad hypothesis. This is these studies are tough. I don't, admittedly, my understanding of them is not as nuanced as the others, and a lot of them come from mouse experiments. There was a promising study of mouse experiments with hemoglobin, a mouse model of sickle cell from a couple of years ago, which suggested that mice with human sickle trait were protected from cerebral malaria, except if you deleted hemoxygenase, which helps to neutralize the heme that's released by hemolyzed red cells. But so this is really exciting data. There was a sort of summary of it that was written in the New England Journal subsequent to, or a month or two later. Subsequently, I've seen some human studies which have suggested that the story is much more complicated than that. And I think that, and Bryce Weinberg from who works at the VA, worked for years on hemoxygenase. On nitric oxide. It's not directly related to hemoglobin S, so, but I think that there could be some sort of shared phenomena between how nitric oxide operates in vivo and how hemoglobinopathies operate in vivo. And adaptive immunity, like I mentioned before, there's no known correlate of immunity in malaria, so these studies are really tough. This is data from 1961 from, I think this was in the Gambia, where children with malaria were treated just with purified immunoglobulins from adults in Kenya, and they essentially cleared their parasites just with antibodies from adults in Kenya, underscoring the fact that antibodies could be very important. Whether or not they're absolutely necessary is a different question. And there are some field evidence to suggest that the pattern of protection in hemoglobin S suggests that it's due to some adaptive immunity. In fact, they titled this paper "An Immune Basis for Protection by the Sickle Cell Trait," but I've never been terribly convinced by it. There are some data that indicate that parasitized red cells with hemoglobin S are cleared out by the immune system more quickly in the spleen and by phagocytes than otherwise. But a lot of these studies have not been replicated, and it's unclear in vivo exactly whether or not that is a important mechanism or if it's just something you show in vitro. So there's a lot of question marks, essentially. There's a lot of unanswered questions. I'm happy to talk about them afterwards if y'all would like. But I'll come back to why I think it's important to study genetic susceptibility to malaria, because like I said, I'm not suggesting that we go try and give children in sub-Saharan Africa sickle cell traits. But we've made this well supported this field studies showing this epidemiologic observation of a quite profound protection from severe malaria, for at least for hemoglobin A, S, and CC. There's some, I think more and more data are pointing to aberrant expression of parasite proteins on the surface of the infected red cell as a mechanism, but ultimately we want to take that forward and find out exactly what is, if not the molecular and cellular mechanisms for that, and hopefully use it as a platform for future interventions to protect these kids. And I'll say that, like I might have mentioned before, that to a certain extent these kids are a natural experiment. If you can imagine that some laboratory researchers come up with a mouse that had a 91% protection from severe mouse models of malaria, they'd be pretty excited about that and really try and drill down to that and understand the molecular mechanism. But these are children who nature has put forward as a natural experiment of protection. So I think that it's not low-hanging fruit, but figure out what the mechanism is, but how you deal with it. So that's Gordian knot has been split. I'll thank all these people. Gary Cox did not bring the tamales as he promised, so I won't thank him. But I really do thank you all for listening, and I hope you all. Thank you.
Thank you.